Symposium both online and offline, faculty and dear students. A very good morning to one and all. I'm Dr. Harita Polinini, Scientist, Division of Genetics, ICR IRI. On behalf of Genetics Club and Indian Society of Genetics and Plant Breeding, I take the proud privilege of welcoming you all to this 3-day symposium being organized under the theme Tending Mendel's Garden for Perpetual and Bountiful Harvest. This symposium is being organized to commemorate the 200th birth anniversary of Professor Gregor John Mendel, the father of genetics, whose experiments on pea plants have been instrumental in establishing the field of modern genetics. At the outset, it gives me immense pleasure to welcoming the dignitaries onto the dais. Esteemed Chairman of the session, Professor R.B. Singh, former Chairman ASRB and former President National Academy of Agricultural Sciences. Chief guest of the session, Dr. Trilochan Mahapatra, Secretary DARE and Director General ICR, who will be joining us online. Dr. G.P. Singh, President, Indian Society of Genetics and Plant Breeding. Dr. R.P. Sharma, President, 15th Genetics Congress Trust. Dr. A.K. Singh, Director and Vice Chancellor, IRI. Dr. Vinod, Professor, Division of Genetics and President, Genetics Club. May I request you all to kindly grace the dais. May I now request our beloved director, Dr. A.K. Singh, to extend a very warm welcome to the dignitaries with a bouquet of flowers and a memento, which is a miniature of the Mendel's bust, as a token of our love and respect. To the chairman, Professor R.B. Singh. To the chief guest, Dr. Trilochan Mahapatra, who joined us online. Dr. GPC, President ISTPP. And Dr. R.P. Sharma, President, 15th Genetics Trust. Thank you, sir. A ray of light is a ray of hope. Let's all leave the darkness and step towards the bright path. May I now request the dignitaries on the dais to light the ceremonial lamp. I would also request the dignitaries to pay respectful homage and floral tributes to the pious soul of Professor Mendel. Thank you, sir. I will now request Dr. A.K. Singh, Director IRA, to present formal welcome address and also introduce the chairman to the House. Sir, please.
A very good morning to each one of you. Honorable Chief Guest of the inaugural session, Secretary Dare and DG ICR, Dr. Tilochan Mahapatra, Professor R.B. Singh, Chairman of the session, Dr. R.P. Sharma, President, 15th Genetics Congress Trust, Dr. G.P. Singh, President, Indian Society of Genetics and Plant Breeding, Dr. Vinod, Professor of Genetics and Organizing Secretary of the Symposium, distinguished delegates, representatives from seed industry, students, ladies and gentlemen. I extend a warm welcome to each one of you to this 3-day National Symposium on Tending Mendel's Garden for a Perpetual and Bountiful Harvest, which is being organized to commemorate birth bicentenary of the father of genetics, Gregor Johann Mendel. The title of symposium, Tending Mendel's Garden for Perpetual and Bountiful Harvest, was suggested by Dr. S.R. Bhatt. Thank you, Dr. Bhatt. You are not here with us. I hope you have joined online. And this has really shaped the entire symposium. This title metamorphically depicts the science of genetics and its importance in future human existence. The symposium is being jointly organized by Genetics Club, Division of Genetics, 15th Genetics Congress Trust, and Indian Society of Genetics and Plant Breeding. We have with us Dr. Tilochan Mahapatra, Secretary Dare and DG ICR, as the chief guest of the session, who will deliver the inaugural address. Thank you, sir, very much for joining us in spite of your not so good health online. We are really indeed pleased with your presence. As you all know, Dr. Mahapatra is a distinguished geneticist and molecular biologist who has made seminal contribution to plant genomics, genome mapping, and molecular breeding. Besides, he is a teacher par excellence and has made phenomenal contribution to human resource development in the field of molecular biology and biotechnology. We welcome you, sir. To chair the session, we have with us Professor R.B. Singh, a distinguished geneticist and former director IRI. His contribution to national and international agriculture is phenomenal, for which he has been bestowed with one of the highest civilian awards, the Padma Bhushan. Sir, we are deeply honored with your gracious presence here on this occasion. We welcome you heartily. We are also blessed with the gracious presence of many distinguished geneticists, breeders, and scientists in the field, offline and in online mode. We have with us Professor P.K. Gupta, Professor R.M. Singh, Professor P.L. Gautam, who have joined online, Professor R.K. Singh, Professor S.L. Mehta, Professor B.D. Singh, Dr. K.V. Prabhu is here with us. Dr. B.M. Prasanna has traveled long distance to be with us. Thank you, Dr. Prasanna, for making it. Then we have Professor N.K. Singh, Professor Akhilesh Tyagi, Professor Balram, Professor Deepak Pental, all have consented to participate in the symposium in the respective session, and the link has been shared. They will be joining online. Dr. A.K. Joshi and Director of Sister Institutes from IRI, Dr. D.K. Yadav, ADGC, Dr. Patil, who is going to be one of the speakers. I also see Dr. Tandon, Dr. Gulati, our teacher, and many more. I welcome each one of you wholeheartedly to this symposium. This symposium is one of the several that are being conducted around the world to celebrate this occasion. This event, we will have gathering of geneticists to reminisce the development of science of genetics along with budding talents from schools and institutions of higher education in the coming 3-day symposium. As a part of this celebration, a bronze bust of Gregor Johann Mendel has been installed in front of Division of Genetics, IRI. I would like to place on record the humble contribution made by the scientists of the Division of Genetics in this great endeavor. Generous contribution from Dr. B.M. Prasanna, Dr. A.K. Joshi, and Dr. B.P. Singh is duly acknowledged. The artistic vision of Dr. K.V. Prabhu and Professor B.D. Singh in shaping the Mendel's bust close to how Mendel's looks like, and that you will see when the bust is unveiled tomorrow at 9 o'clock. We have planned, you will realize that a great job has been done by all collectively. The bust is sculpted by a famous sculptor, Padma Shri, Professor Biman Bihari Das, and this will be unveiled on 20th July 2022. We are sure Mendel's bust will instill inspiration for the generation of young scientists, geneticists, students who come here to learn science of genetics. I once again welcome each one of you wholeheartedly to this symposium. Thank you very much.
Thank you, sir. May I now request our co-organizer, Dr. G.P. Singh, President, Indian Society of Genetics and Plant Breeding, to address the delegates.
Honorable Secretary Dear and DG ICR, Dr. T. Mahapatra Saab, Professor R.B. Singh Saab, Dr. R.P. Sharma ji, Dr. A.K. Singh, Director IRI, Dr. Vinod, Secretary of the Genetics Club, the Division of Genetics, galaxy of scientists who have assembled today in this hall and also on online mode, Professor P.K. Gupta Saab, Professor R.M. Singh, my teacher, Dr. B. Mishra, Dr. B.M. Prasanna, Professor N.K. Singh, Dr. K.V. Prabhu Saab, Dr. Tandon, colleagues from ICR, Dr. Shashni, Dr. Raman, distinguished guest here, scientists, students, ladies and gentlemen. I would like to take this opportunity to thank respected Director of the IRI, Dr. A.K. Singh, who have thought of organizing this kind of symposium on the bicentenary celebrations of Gregor Johann Mendel. In fact, when we also thought of from Indian Society of Genetics and Plant Breeding holding such kinds of celebrations for bicentenary celebrations, I came to know that IRI is doing it, and I contacted Dr. A.K. Singh, and he very readily agreed to involve us as a co-organizer of this symposium. I think the program of this symposium has been very meticulously planned, and I have seen during my past experience, 30 years, probably the students who come now for graduation in genetics or plant breeding probably talk of very high throughput science, but are forgetting a Mendelian inheritance, and therefore it will be a good opportunity to recall, the contributions of Gregor Johann Mendel made in 1865 when he presented his paper, which led to the development of Mendelian inheritance. As it happens, the newer things were opposed, and at that time, many of the people did not agree to what Mendel said at that time. But later on, in 1900, the rediscovery of Mendelism took place, and this led to several of the discoveries, new thoughts, new philosophies. and the results are evident throughout the world, not only in agricultural science, but also in medical science, microbial genetics, and so on and so forth. So this is a very good opportunity on behalf of Indian Society of Genetics and Plant Breeding, the Executive Council of the Indian Society of Genetics and Plant Breeding, and on behalf of the Office of the President of the Indian Society of Genetics and Plant Breeding. I thank you, sir, for giving us this opportunity to be a part of this symposium. And I hope this will be a grand success, not only for colleagues like us and our elders, our teachers, but also for the students who are enrolled new in this field of science. Thank you very much.
Thank you, sir. May I also request Dr. R.P. Sharma, President, 15th Genetics Congress Trust, to address the delegates. Sir.
Good morning to everyone. Professor R.B. Singh, Dr. Mahapatra, Dr. G.P. Singh, Dr. A.K. Singh, Dr. Vinod, the dais, Professor P.K. Gupta, Dr. Prasanna, Dr. N.K. Singh, Dr. R.N. Singh, Dr. Prabhu, Dr. Tandon, Dr. B.D. Singh, Dr. S.S. Patil, and others distinguished colleagues and friends. It gives me immense pleasure to be participating along with you in the celebrations to commemorate bicentenary year, birth century of Gregor Mendel. When I stand here and share with you my views, I go back to the year 1962 when I joined this institute, and Dr. Swaminathan asked me to work on Drosophila. And from that, actually from there onwards, I got associated with genetics. I used to teach genetics 100 practicals, and many of my colleagues here, they know how interesting it used to be. Because when I joined and I was given the responsibility to teach genetics 100, I was also handed over a few packets of maize kernels, colored with different colors and mixed together. And we used to ask PhD students to separate them into different colors and apply Mendel's segregation ratio. That used to be the teaching of genetics in IRI. But subsequently, when we started with Drosophila, then we actually started doing Mendel's experiments. Many of our colleagues, Professor P.K. Gupta, I remember, he acknowledges also, he used to take Drosophila culture from us. J.B. Chaudhary from HAU, Professor Akshay Gupta from PAU, and other places, Delhi University, also some people, they used to collect the cultures from us to teach Mendel's laws. Well, this is the history behind it. The credit actually for populating genetics in IRI solely goes to Professor Swaminathan. When I joined it in division of botany. There was no genetics division. Genetics division commenced only in 1966. He was, and he is such a visionary fellow, and all of you know because you have come in his contact. Being an agricultural university, the institute, he allowed to work not only on Drosophila, on E. coli, on human chromosomes. In fact, to cover the various facets of genetics which need to be covered. And that was the beauty. And I hope that that tradition is still being continued. Agriculture is not a separate subject. Biology In fact, genetics encompasses all the components, all the subjects in biology. And that's the beauty. The moment you start with genetics, you forget about distinction between different organisms. They are same. Basically, they are same. The principles remains the same. I am today here standing as president of the 15th International Genetics Congress Trust. It's a coincidence Dr. Swaminathan should have been here, but because of his age, next month on 7th August, he will be entering in the 98th year of his life. He's weak, but he is still very active mentally. In 1983, we organized Genetics Congress in International Genetics Congress, 15th International Genetics Congress in New Delhi. The function took place at Ashoka Hotel. And this is his vision. Many people would realize how, at that time in 1983, after executing the responsibilities of the Congress, there's a certain amount of money which was left. And he and Professor Chopra, both of them, they took the decision that this money we will invest into a trust, and the trust will be utilized for propagating and disseminating genetics knowledge in the country. In the country for the students, in the country for the society. And that's what precisely we have been doing now in the trust. In the last session, which is devoted to teaching in education, I will elaborate it further, but sufficient to say that we are doing these experiments in last few years. You will be surprised to know more than 200 teachers of Delhi public and private schools. They have been exposed on hand-on experience on various protocols, experimental protocols in genetics. We take them to the institutes in Delhi, where people are working in those areas, and make them understand what exactly it is. It is turning out a very, very interesting experiment, because most of the teachers, they used to say that we understand, we read from the books, but our concepts are not clear. And that's why we took the decision that our trust will take the responsibility, take the teachers to the labs and ask them to do with their own hands the experiments which they need to be done. And that's what we have been doing. I'm enjoying it. I would like to thank all of you for your patience, and thank you very much. I give success to A.K. Singh for organizing this meeting. In fact, 1965, I remember Mendel's work we were commemorating, and the symposium took place in this hall. And I remember standing here in 1965 with many international luminaries and giving a talk on Drosophila indirect effect of radiation. So that's the time which has passed through over almost 60 years or so. But it has been fantastic. And I'm commemorating such people. You will in next 2 days, all you know, because you are all students of genetics. But I think it's always good. It gives you, it satisfies you to remember such luminaries who have really provided completely new insight. The whole biology world, in fact, not even biology, even the physical sciences, have completely transformed the world of life. And that's where we are enjoying Mendel's Bicentenary. Thank you very much. Thanks a lot.
Thank you very much, sir, for sharing beautiful memories. We now have 2 publications awaiting their release in the symposium. May I now request the dignitaries on the dais to launch these publications for the wider benefit of the audience readers. First one is Book of Abstracts, compiled and published by the Symposium Secretariat under the supervision of the organizing chairman, Dr. A.K. Singh, Director, IRI. Next, we have a book entitled Fundamentals of Field Crop Breeding, Volume 1 and 2, edited by Dr. D.K. Yadava, ADG Seeds, Dr. H.K. Dikshit, Dr. G.P. Mishra, and Dr. Shailesh Tripathi. May I request the editors to kindly come on to the dais, please. Thank you, sir. May I now request the Honorable Chairman of the session, Dr. R.B. Singh, to formally introduce and invite the Chief Guest for the inaugural address.
A very good morning to each one of you, friends, ladies and gentlemen. The Honorable Chief Guest, Professor Trilochan Mahapatra, most illustrious Director of this Premier Institute, Professor A.K. Singh, Secretary of the Genetics Club, Professor Vinod, the Chairman of the ISGP, G.P. Singh, R.P. Sharma ji, who's the President of the 15th ISG, Genetics Congress Trust, is that right, sir? Congress Trust. Outstanding colleagues who are really present here. Fellow geneticists, I see so many of you. Maybe if I can try to read at least the frontline names, if I remember. If I remember Patil Sahab, is that correct, sir? Professor Gupta ji, Varmari Mishra, of course, Prasanna ji, N.K., Rishi, Vinod, Prabhu ji, Tandon Sahab, आपका नाम मैं भूल रहा हूँ. And of course, B.V. Singh ji and many other colleagues. Ladies and gentlemen, dear fellow students, I must congratulate the organizers and the co-organizers who have really done this landmark event and are really celebrating, which is being celebrated all over the world, as a matter of fact, this bicentenary occasion of Mendel's birth. As a matter of fact, in 1965, when the first, of course, the anniversary of publication or the presentation of Mendel came in this very hall, as Professor Sharma mentioned, we had Mendel Symposium again. And that was the symposium which was attended again by the world famous scientists. I remember my own professor from North Carolina, Professor Robinson, H.F. Robinson, the father of the development of the new breeding technologies and so on. Robinson, Comstock, Harvey, many of these names must be familiar to you. They were here. Professor Munsing was here. Professor Gustafsson was here. You can remember the kind of attraction this very hall had in 1965 to celebrate that 1965, which was the celebration of 100 years of the presentation of the Mendel's work. Amazing. And again, we are doing this at such an occasion. This is really indeed a tremendous tribute to this institute, which has been doing it time and again showing to the whole world its dues and genetics. Friends, genetics is the science of thread of life, whose father is none other than Gregor Johann Mendel, who is one of the greatest gift to the planet Earth ever given by any resource. We are very fortunate on this occasion to have an outstanding person who is not lesser than Professor Mahapatra to really enlighten us on genetics and sustainable food security in India. I need not introduce Professor Mahapatra in detail because I will always feel short of words in adequately describing his achievements. But let me take pride in mentioning that Professor Mahapatra was a MSc and PhD student of this great institution, did his PhD in genetics from here in 1992. He has been the most illustrious secretary of DARE and the Director General of the ICR. Prior to this, as you all know, he was the Director of IRI, and before that, he was Director of CRRI, Cuttack. As a matter of fact, I had the privilege of working with him at all these places in one way or the other. And as a matter of fact, we were able to see together that how the best of the systems could really operate and be progressing their work are based on Mendelian genetics. He is a world-renowned molecular geneticist. And today, when we talk of Mendel's laws, Mendelian genetics, of course, you have to link it with the molecular genetics and understand the connectivity right from the word genetics was coined and right from any of those developments of the three laws of Mendel were brought to bear upon the development of agriculture, or for that purpose, any science related with biology. Among his sterling research contributions, Professor Mahapatra was one of the very first one to have developed the high-yielding basmati rice varieties resistant to bacterial blight, the biotic resistance that he was able to bring in, the genome sequencing of rice, tomato, and so on. As a matter of fact, he was also responsible for a large number of the developments of the human resources groups that we have here in this hall. Dr. Mahapatra was considered to be one of the best teachers. In his classrooms, chairs will not be enough. Very often students will stand and listen to him because he was such an attractive teacher and he continued to be that. He has been decorated with a large number of awards and honors. Of course, the INSA Young Scientist Award, LSS Kumar Memorial Award. We all know that many other Prakash Machine Award, the NAS Tata Award, Lifetime Achievement Award of so and so. He is fellow of INSA, NASI and NAS. Of course, as you all know, he is the president of the National Academy of Agricultural Sciences. He has received honoris causa degree. I can't count them on fingers on so many, from so many universities. As a matter of fact, my dear friends, I don't want to stand too long between him and you because I know you are eager to listen to him because he is the rare speaker. Professor Mahapatra, I have the privilege of inviting you to kindly deliver your lecture. Your Thank you.
Thank you very much, sir. Thank you very much, sir, for your kind words of introduction. My voice is choked, so I don't know how much I'll be able to speak. So, so can you hear me? Can you hear me?
Yes, sir, we can.
Yes, sir, you are audible, clear.
Okay. Okay. So I can't have more volume than what I am doing at this point in time. Might reduce further. So, Dr. Arbis Singh has used so many adjectives, and that is his usual way of giving importance to others. He is a wonderful human being, so respectful even to the juniors and students, and always so encouraging and inspiring to hear him. Your kind words are always of great value to us, to me particularly. So whatever little I have been able to do, it was all in collaboration, because that's what I strongly believed, that from the beginning, that we must work in a school format, and we should be interacting to deliver on the targets. Collaboration, cooperation are the cornerstone of success, and very importantly, doing something which is time bound and has to be with respect to solving a particular problem. So given that background, so thank you very much, Professor RBC. Thanks to Dr. A.K. Singh for inviting. So I was extremely happy and readily agreed that I should be here at this occasion as a student of genetics to celebrate the personality like Gregor Johann Mendel, whom we worship, who laid the foundation of genetics. And as a student of genetics at IRI, it was still very exciting to learn the fundamentals of genetics through Mendel's work. And of course, that excitement one would never forget in life. You need teachers to actually teach Mendelian genetics, and to, in the process, to excite others. And this is the lesson we learned at IRI, and how important it is to put the proper perspective with regard to Mendelian concepts, bringing in theory, practice together, and then in the process, enlightening students to get the essence of the science of genetics, how it was conceived, and how profound it has been with regard to its use. Impacts are quite large. Of course, Gregor Mendel's work still remains relevant, and we continue utilizing the same principles in our day-to-day work in the arena of plant breeding, the crop improvement. Gregor Mendel's courage and persistence to do something is very well known. The intelligent way of dealing with complexities, and in fact eliminating those which don't follow is the standard path which he thought appropriate, was most crucial for his success. Often as investigators, as scientists, we get embroiled in complexities fast, try to resolve those, and in the process, we get lost. We don't discover anything new. And that was the kind of intelligent and strategic way of handling. Is a crop, is a process, is a progeny, and segregation of progeny, and interpretation thereof. Each one of them provide enough opportunity to learn and enrich ourselves. The concept, though is not very well defined, the very fact that you use those symbols, capital and small, to describe the elements, although the chromosomal theory comes later. But in the process of using those symbols, very clearly illustrating the allelic concepts, that duality of the gene, duality of forms of gene, which was so well described. And that was something which was not very apparent when he talks, when he describes, but so clearly that emerges. And subsequently, we went on using mutagens to generate large number of alleles for various traits or different genes. And by way of using capital and small letters, again very intelligently, he depicted the dominance and recessiveness of those alternative forms. Again, another intelligent way of expressing totally unknown concept and alluding to the fact that there is existence of dominance. And of course, the recessivity. The appearance of recessive ones in undiluted format again says that Mendel was very clear, and his mathematics, statistics, whatever he understood, that was handy in explaining the ratios of the recessive homozygous ones which appear as compared to the dominant genotypic forms and the alleles in combination of homozygosity or heterozygosity. So why I'm talking about this is that while we are remembering Mendel today on his bicentenary, it's always exciting to go through what he has actually given in his long paper, which he presented. Today, presentations are not considered very important. The citations that are received by presentations are not many. So presentations which are thoroughly, in fact, investigated based on thorough investigation, years of investigation, so are also equally important. And that context, our symposia are to be actually redefined and redesigned to put a equal emphasis on oral presentations as well as poster presentations. And they speak volumes about what is actually, depicted and what was done. In fact, Kerry Mully's work when he presented before Joshua Luttenberg and tried to explain him and what it is, he only shook his head and went off without any comments. So often one has to face that kind of situation. Nobel Prize winning work, not really getting adequate response from people who matter. And one gets disheartened in the process. So Gregor Mendel has gone through all that, the ignominy and all that was quite at its height. So working despite adversity and giving something new, for which we are worshiping him today as the father of genetics, cutting across kingdoms, whether it is plant or animal or Drosophila or other systems, whether C. elegans. So every system has developed model organisms, and those models provide plenty of opportunity for us to learn and again revisit Mendel. Despite all his contributions and subsequent to his beautiful description of genetics and its myriad facets of implications, we understand how and why the offspring is related to the parents, parental lines. We understand why children resemble their parents, and that because of the genetics. And Mendel very clearly established this. And it was also very clear, although it came much later, that they come in pairs. Capital A, small a, not only they depict two different allelic forms, one dominant, one recessive, but they also remain in pairs. And chromosome pairing behavior, and chromosome as a carrier of genes. And we know when it came and how it was proved. And very clearly, genes don't blame. The alternate forms don't get diluted. The appearance of recessive forms clearly depicted that. Again, a very exciting thing to observe and also carry home back to use. And of course, some are dominant, some are recessive, some of those forms. And very clearly he said that there are rules which are followed. Rules which are followed. So the law of segregation, law of independent assortment. So rules are followed in biology and using mathematics to explain that. So how elegant he was, how clear in his mind about what he was doing and trying to explain. And very importantly, genes are real. The elements are real. They are not abstract. And that was proven subsequently. So That's very, very important message that we get from his work. And the pre-existing ones gives rise to the new ones. And that is something which is very, very important. That something new doesn't come, only pre-existing ones reappear. And that is a message, but we are in today's concept and developments, we do find newer developments and ideas, new science coming, which deviates from what was actually described. Of course, a number of them which came later. The sex-related chromosomes, sex chromosomes, and so on. That chromosomes carry genes. So he didn't describe this, but certainly that is another milestone. The very fact of assortment, independent assortment, tells us that the reshuffling is happening. And that reshuffling and how it happens, when it happens, and what are the deviations, certainly took several decades to prove all this. But certainly he said that that is reshuffling taking place. And of course, the evolution theory of Darwin that described elegantly the genetic basis is actually clearly, with evidence, demonstrated by Gregor Mendel's work. And of course, subsequent works. Of course, it is true that all that we see in nature cannot be explained by Mendelian principles, theories, and laws. Though these are all accepted, proven, but the deviations are too many. Deviations are too many. So I will not go further in describing Mendel and his work. And for youngsters, I thought it would be worthwhile revisiting what Mendel said and how relevant they are. Though Mendel's paper in original form is not taught in many universities, only the law of segregation, law of independent assortment, and some books include law of dominance as a separate law. And those are described mechanistically, and not much of insight one finds with regard to the philosophy of it. And in original paper, if one goes through critically, one finds a number of things there, including heterosis. So how they are actually described. So if one really goes word by word and line by line, and I urge today's teachers of genetics not to just go by books and chapters which are there. Of course, the books by Dr. B.D. Singh, Dr. P.K. Gupta, they are quite well written, and students are actually using those books, very cheap and very well illustrated. So I believe for teachers to teach, they must go through the original paper that was presented by Gregor Mendel. And so many ideas one would get in addition to the intelligent way of handling material and the persistence one gets. And long years, 8 years of experimentation. Our results are published 1 year or 2 years of experimentation. And more than 3 years, we don't work even for our PhD. We don't have patience to actually work. So there are shortcuts. So I believe that these are the aspects which we need to really deliberate on occasions like this. Professor R.P. Sharma is here, a great teacher who taught me the fundamentals of molecular genetics. I am grateful to you, sir, for hand-holding, for mentoring, and your work on Drosophila and its genetics, using mutations, and recognized globally. And that's the kind of dedication that is always, one has to emulate and follow. So that is the kind of lessons we learn from our teachers, a kind of not just classroom teaching and what is there in books or journals. More than that, teachers teach more than what is there in this. And they are the teachers who actually give lessons beyond what is there in books and literature. And those messages are career-building messages, life-enriching lessons and messages. So thank you, Professor Sharma, for all those contributions that you are here today. And we are grateful. Of course, Mendelian principles have been utilized for crop improvement. And obviously, the global hunger, malnutrition, and all that prevailed in the previous century, and that we see this century, these certainly our scientific fraternity have tried to address by using the fundamental principles of genetics as outlined by Mendel, and subsequently enriched by various other experiments, including experiments on mutations and experiments on various other facets of genetics. Discovery of DNA as the genetic material and double helical structure of DNA and many others, gene-protein relationship and enzymes and role of enzymes and so on and so forth. Every single development has contributed to development in science. Green evolution happened because of use of Mendelian principles. The famous RST locus in wheat giving semi-dwarfing phenotype is a case to mention that so elegantly brought green revolution. How that particular gene and other alleles of that gene, how they were utilized to build material and in the process distribute those, enhance crop productivity and eliminate hunger to a large extent. So these are all examples before us. And similarly, DGO gen, the semi-dwarfing gene in case of rice. The Taichung native 1 contributing to IR8, and in India contributing to Jaya, Padma, Ratna, subsequently number of varieties, which giving us the revolution in food production. The RST, including DELA protein that we know today, and how it influences the elongation, stem elongation. I strongly believe the GA signaling pathway. I'll not go into details, because if I go into details of each one of those, that are how genetics and food self-sufficiency has been gone hand in hand, and contributed to not just green evolution, but subsequently even other revolutions that we see. The kind of protein, kind of alleles that we see today, and variation within gene RST, giving multiple forms with varied level of expression, GA non-responsiveness even brought out very clearly in certain cases, and alternate forms of RST or SD1 in case of rice. All these point out to the fact that nature has a plenty, and there is plenty to exploit with regard to Mendelian genetics and Mendelian principles. Those alternative forms are there at large, undiscovered or exploited in the germplasm. Millions of germplasm sitting there in our gene banks. One estimate says that not even 5% of the germplasm is utilized even today. So the Mendelian principles to work and continue contributing to the revolutions in agriculture, in crop agriculture or horticultural revolution or crop revolution. I believe there is plenty of opportunity for us to harness unexploited genes and alleles. Allele discovery is more crucial and critical than even gene discovery. If we say that even RST, B1B, D1B, and even a whole lot of others, how they express and respond to GA, and what kind of effect they have, or SD1 and the alternate forms, what they really do and how they work. It's actually deep science. And certainly, we should be able to bring out those from the germplasm to actually build new revolutions. Alternative forms might also give additional traits which were not available in the original form which was discovered in the form of wild type. And there lies the importance of creating more mutations, even those non-existent ones created. And there comes the role of chemical mutagenesis, which was forgotten. And even today, we don't take it seriously, because quite a bit of work, Professor R.P. Sharma's leadership, again, we initiated the chemical mutagenesis in Agina 22 in rice and created alternate forms. We have seen its seed size gene. And in that, how mutations at different locations create many different effects on seed size. Same gene, different mutant sites, one truncating the protein, the other leading to amino acid substitutions, missense mutation. and different locations of those substitutions, and in the process, seed size greatly varying. That work is now published. And this is a beautiful example. And also, we sequence in germplasm some of these genes to discover haplotypes. And some of these haplotypes, which was published way back in 2008. And these haplotypes are so different. Combination of S and Ps in the same gene, giving different proteins. That is what we learned through modeling, model building. And those proteins having different kind of interactions, and the process contributing to different expression of the trait. So I'm only focusing on one, a Mendelian gene, a single gene, and different forms, alternative forms of the same gene, and contributing differently to the trait expression. And some of those expressions are highly beneficial with regard to gain of trait. In nature, what we see and what we subsequently described as quantitative trait locus. When as a student in 80s, I could gather based on isozyme studies in maize. Charles Tuber's work, Dr. Prasanna is there, who remember those, and delivered the seminar based on Charles Tuber's studies and Edward studies in maize using isozymes, how QTLs were mapped. Mendelization of, in fact, traits with complex expressions, phenotypic expressions, was unthinkable in 1980s. And our own teacher, who used to teach quantitative genetics, was of course was not accepting that this is possible. So counter theories were actually put forth, and accepting those initial work was very difficult for those who believed in abstractness of those factors or elements which contribute to the quantitative trait expressions. The statistical entities and the statistical expressions putting abstractness at the core, probably that was the kind of concept then, and breaking into those kind of hard surface was not easy. Globally, so Mendelization of those complex traits, breaking them down into even major loci, which contribute and even to the extent of 60% of the trait expression, explained by a single locus, to a large extent as a Mendelian trait, and even multiple mutations in those loci, contributing to the quantitative nature of trait expression in a Mendelian gene. May not be truly Mendelian factor, but physically, yes, it is the same Mendelian factor. So today we understand quantitative trait loci more elegantly, in a very elaborate way, and cloning of quantitative locus was a milestone. Number of examples are there. I'll not go into those details. The map-based cloning, starting with PTO in tomato, XA21 in rice, and subsequently a plethora of genes getting cloned through map-based cloning approach, including quantitative trait loci. That threw light on the Mendelian factors, but not behaving same way as Mendel observed, but contributing to revolutions. I'm sure what we have today in terms of agricultural production, the credit goes to Mendelian genetics. And of course, quantitative genetics, not the theory part of it, which did not contribute much, but the practical part of it. And that is largely the Mendelian principle of doing crosses, going to the progeny, selecting, and then going back and crossing, and further selecting, and largely following the Mendelian route. We have today a satisfying position with regard to food grain production. It's already 11:17, and I'll take maybe another few minutes. What is that we have today? 1950-51, if you compare, we have more than 6.5 times increase in production. When I say times, that's 600% increase in production in India with regard to food grain. So that's not a small achievement. Of course, it doesn't happen overnight. The grain revolution happened, but that was not really enough. It required many, many efforts subsequently to sustain that. And in the process, build buffer stocks in rice and wheat, which can be actually utilized to feed more than 80 crore people for months, more than 6 months rather. And that continues even today during COVID times. How it was possible? It's all the miracles of genetics. Today we have not only buffer stocks there, but also we are inching very close to self-sufficiency in pulses. I must say, while breeding on one hand is important, using principle of genetics is important, and level of variety is important. That's not really enough to have these kind of changes that we have seen in production front. In case of pulses, we are hovering around 15-16 million tons before 2015-16. We had varieties, new varieties were not going to feed. The old varieties, seed replacement was not happening. Farmers were not interested in growing pulses because the MSP was not really well remunerated. Government didn't have any policy of procurement for filling the buffer stock. All these policy decisions were taken. That one would go for procurement, at least 25%. Create 2 million stocks, 2 million tons of buffer stock, and procure from the farmers. Simultaneously, what was planned is indenting new varieties only. The old varieties indent were not accepted. 150 seed hubs were created to produce seeds and provide to the farmers the quality seed. That led to variety replacement to the tune of 80% in pulses, unthinkable. Seed replacement going beyond 30%, which was not even 10% in most pulses before 1450. So you can keep applying Mendelian principles or genetics the way you like, but you cannot bring revolutions in production by way of productivity increase unless there is the right kind of policy in place and unless the technology goes to the field. And this is what the example of pulses revolution. Not many actually describe this pulses revolution. Why? Because there is no foreigner to tell that, look, India has a revolution in pulses because we worship foreigners. That is the tendency we have. A gold or somebody else, some person should come and tell us that, look, you have a pulse revolution. And we will actually, like a parrot, will repeat what he has said or she has said. You have to really set that kind of attitude. 2015, 14-15 or 15-16, what was the production? 17 million tons. And 21-22, what is the production? 27 million tons. 10 million tons jump and sustaining from 6 million tons, first year jump to 10 million tons jump, sustaining during this period of 6-7 years. And in fact, very little import we want, that too only in certain extent to in case of lentil and in case of pigeon pea. Very little. Import substitution has happened in a very significant way. Our textbooks must contain a chapter on pulses revolution. What it is, policies are barren without technology. I keep telling this. I told this in the recent meeting of honorable ministers of agriculture of this country, who gathered in Bangalore from various states under the chairmanship of Minister of Agriculture, Government of India. And this meeting, I said, this very statement that the policies are barren without technology. And technology is very much handicapped without right kind of policies. But also, I must mention, a disruptive technology does not require any policy formulation. It drives itself. Do we have that kind of technology? And how many of those disruptive technologies are available with us? We have one BT, Cry1AC, Cry2AB, and few other genes to bank open the whole transgenic development, GM. Why we are not moving? Globally, where is the investment, the private and public both? What are the new genes? Past 10 years, we don't see. What are those new genes similar to BT genes bringing revolution in the GM field? The same old glyphosate resistance and BT insect resistance, a few of those old genes are still running here and there. So, where is gene discovery happening? traditional way and modern way. Conventionally, are we bringing revolutions by way of discovering genes similar to semi-dwarfing ones, having profound effects on plant architecture? We keep working on plant architecture genes and traits, but without any significant actual contributions. They're all incremental contributions. 5% is the margin that we have in all India coordinated trials. Often we don't cross them. And we only try to stabilize by way of putting some resistant genes here and there, which are well characterized, except that there are no such genes, to my knowledge. In the past 6 years, I'm here in this position. I don't see any such development which would have a quantum jump in productivity and bringing that kind of revolutionary change. More so in pulses and oilseeds. That is where we need. While I was trying to talk about and mention and describe pulses revolution in India, 10 million tons of more production, new varieties going to the field, replacing the old ones. So policy, technology going together. So technology is to be taken to the field. That is another aspect of what I was telling. But I did mention, and I'm trying to recapitulate so that everybody goes with this message, that while working on one hand, lab to land, Honorable Prime Minister keeps talking about it, emphasizing on this. We believe that we will have some demonstrations and it would work. But that's not very effective in today's context. We have not been able to really succeed in many regions of the country with regard to demonstrations. Our frontline demonstrations gave 50 to 100% more yield than the farmer's field performance. But how much of them are accepted? In many crops, our yield is low, not because we don't have varieties. Genetics has played its part, but it's low because management is not happening. I keep telling this, that if Punjab wheat yield is 5 tons per hectare, why it has to be 1.5 tons per hectare in Chhattisgarh? Should we really go for this? Are we really deploying genetics to defeat ourselves? Genetics has to win for us. Genetics cannot defeat us. So if we have varieties which give 5 tons average yield in Punjab, and 7 tons and 8 tons we are getting. Now we have varieties, Dr. Joshi would be there from CIMET, and every year we raise the bar, and we should have 8 tons, 9 tons, and now 10 tons. And we have some varieties giving more than 8 tons in farmer's field. So that's all genetics. Put a target, work systematically in that direction, collaborate, don't work in isolation. Use the vast genetic diversity which is existing. Use global diversity to the process of this partnership and build material. So that is what is very, very important to harness the full benefit of genetics. So while we are building this, we must not lose sight of working in the direction of taking to the field. So colleagues, there are many things to talk about. Two more areas, so I'll briefly touch upon. And then end my lecture. My voice is not still opening up properly. It's choked and getting more and more troubled by speaking. But while actually emphasizing on genetics and creating mutations, I did highlight Professor R.P. Sharma, his work on Drosophila, the wingless mutant, which Professor Sharma created, is a global sensation. How it works in various pathways in a human. And he could have actually won Nobel Prize, with his continuation of work in India. We get leads, we develop something, do something, then branch out to something else. Don't really devote lifetime on those works. And Drosophila work at the genetics division still continues. But it doesn't have that kind of teeth as he used to have. Many Drosophila labs are closed down. And I'm sure, on Dr. A.K. Singh's hub, if we can actually put forth a proposal to revive this in universities where genetics teaching is taking place. Let's have the Drosophila labs and the Maze Corp, Dr. I believe Firoz Hussein's work. It was published in our own Indian Journal of Genetics. How Maze Corp can be utilized in teaching. And if we can create these kits and stocks and provide to these universities to teach genetics, a standard genetics is very, very important. And education is very, very important. So Dr. Sharma, through Genetics Congress Trust, is teaching teachers of the schools, exposing them to genetics. It's much more to be done. And there is a proposal through our education division. We can support, and through National Heritage Education Project, in fact, we can create these labs, support these labs for posterity, so that genetics is revived. So this could be a very good action plan from today's deliberations. And we should be able to actually do this, Dr. A.K. Singh sir. To go deeper, these mutants which we created in case of Nagina 22 by EMS has given us a mutant which is herbicide tolerance, which gives us herbicide tolerance, acetolactate synthase, mutation in that. Imesthaper is the herbicide. Against that, it has resistance, it has tolerance. Dr. A.K. Singh has transferred that to various basmati background, including Pusa Basmati 1121, which still continues to rule. Thanks to Dr. B.P. Singh, I don't know whether he is here today, for creating this marvel called 1121, the longest grain after cooking in the world, the first ever in the Limca Book of Records, attracting consumers' attention globally, particularly in the Gulf countries, earning us more than 19,000, 20,000 crores annually, as against the 18,000 crores, 18,500 crores of DARE and ICR budget. I keep citing this example. This is a beautiful example of genetics bringing a revolution, enriching farmers' income, strengthening country's economy, winning global trade, and a whole lot of positives. Societal impacts are unfathomable, not yet done. Dr. A.K. Singh Saab, I have told earlier, IRI first engaged its economists, colleagues to do an elaborate socio-economic impact of PUSA 1121. It is just not 19,000 crores. It could be 90,000 crores or even 190,000 crores. Who knows what is the social impact of it? We need to actually do this. So this is example of revolution and genetics contributing to this revolution. And today, 4 varieties of basmati of IRI earning for us in the country by way of export more than 25,000 crores. Out of 30,000 crores that we earn, 90-95% are from these 4 varieties. What a contribution of genetics. The conventional breeding, Mendelian principles applied, genes brought together, and this case genes for grain, shape, size, and aroma and quality of the grains, the cooking quality brought together. Another revolution I keep talking about is in sugarcane. How genetics has brought this revolution, and how our friend Harish Damodaran has actually elaborately documented this. Harish Damodaran from Indian Express. Excellent documentation of contribution of the variety CO0238. 20 tons more cane yield per hectare. 2% more sugar recovery. Sugar production increasing by 6 to 8 million tons. UP becoming #1 state in sugar production. Government of India taking decision to divert sugar or sugarcane juice for bioethanol production. Sugar getting exported from India and enabling India in energy security. 10% substitution of petrol by bioethanol. The largest contribution is from sugar and sugarcane juice. And the variety CO238 contributing to this. How do we actually capitalize on all these developments? There are recent examples, but we need to replicate these in pulses and oilseeds. We need to really put our minds together, all the ingenuity at our disposal to have that kind of revolutions in pulses and oilseeds. Not incremental changes, but a jump, a very significant jump in this. Can we employ chemical mutagenesis to do this? And to what extent? I strongly believe there is plenty of opportunity for even chemical mutagenesis to work. Can we not employ gene editing in this particular area? Thanks to Government of India, Honorable Prime Minister in particular, and Honorable Cabinet Secretary, and other officials in the PMO, who actually spearheaded this, helped us to bring out notification. Free gene-edited product, particularly SDN1 and SDN2, from the clutches of the regulatory processes. The regulatory framework was actually throttling, killing the whole gene editing work. We have seen in case of rice that some of these genes, for instance, DST, which was known which was cloned through map-based cloning approach. It was KEMS-induced mutation, giving salt and drought resistance. And now, gene editing in this particular gene at specific sites give us very significant difference in the variety M21010 with regard to drought and salt tolerance. Dr. Bicentenary IRI has developed this variety. How genetics can bring revolutions. M21010 is a popular rice variety. And similarly, there are other examples. The teosinte branched gene, TB1. In rice, it has been mutated by gene editing by ICGB, funded by the National Fund Program of ICAR. Increasing grain number per panicle, number of panicles per plant, and enhancing yield in the process to the tune of 60%. But concomitantly, there is a negative effect with regard to strength of the stem, because the panicle is heavy, there is lodging. May not be much use later, or we would like to have other genes to strengthen the stems. There are other genes known now which can strengthen the stem. And we should be able to harness this TB1 mutation using gene editing to full benefit. The other one we knew that GN1A gene and mutation in this gene, particularly in M21010, and again enhanced yield to the tune of 20%, and grain number to the tune of 40% in M21010. And it is known that GN1A gene enhances grain seed number. And how we can mainstream gene editing now by way of mutating using SDN1, SDN2 approaches. Create as many mutations as possible. Create alleles at will. And that can be done in case of pulses and oil seeds. Another gene, ideal plant architecture, IPA1, again gives grain yield to the tune of 64% increase in rice. And that was in the variety Swarna. Mind you, Swarna is a mega variety. And in certain situations gives 8 tons per hectare yield. Even if we have under field conditions 20% more yield in, over and above Swarna. By way of IPA gene mutation, it will be a revolution. It will be a revolution, a disruptive change. I strongly believe that we should be able to capitalize on this. Our famous Samba Masuri, PPT 5204, we actually supported mutation through ICR program, and mutation again in GN1A, grain number gene. And in this case again, we have very, very significant jump in grain number. I'm sure in another 2 years' time, we should be able to bring very, very significant change in rice productivity in these mega varieties, M21010, Swarna, and PPT 5204, which are so popular. Single gene changes and mutations at specific sites by gene editing and deregulated system, thanks to the government, and proper guideline in place, and SOP should be in place shortly, and we should be able to take it to the field and release them. Can we actually take these to oil sheets and pulses? What we need to really do in oil sheets and pulses? Can we change the grain number per pod in mustard? Instead of 15, 16 seeds per pod, can we increase it to 40 in mustard, Brassica juncea? We have Brassica rapa, where there could be 40, 50, even 60 seeds per pod. The size is small. Can we also increase the size concomitantly? Some genes in Arabidopsis are known. I believe genetics should come to our rescue. We should be able to increase this significantly. Can we have appropriate plant architecture in case of PGNP? Why not? Branching from the base. In Brassica, we have done this. Simultaneous flowering and determinate flowering so that we can have mechanical harvesting. I'm sure we should be able to. We have some early types with a determinate habit in IRI, but they are not very high yielders. I'm sure we can have a little bit of more duration, about 130 days, 135 days of duration. But we need, in case of PGNP, 3 tons to 4 tons yield. Why can't we do this? I'm sure we should be able to really do this. So this is how genetics should play a major role, and in the process bring revolutions in areas, in crops where we are lagging behind. But there is much beyond genetics today. There will be last 5 minutes of my deliberation. They're already going to be, it's 11:50 or so. I'll finish by 12 o'clock. I'm sure there will be time. But it's not genetics all alone. Today's world has plenty of opportunity to understand the nurture component of nature. Nature and nurture components are there from Batson time, even from Darwin's time. And Batson placed this in proper perspective, and that we continue discussing about nurture today. Nurture and nature. When we do actually the debate, we do find that nature, that the genes and the genetic makeup has the fundamental influence on individuals. But nature, the nurture, having overpowering effect on the nature, that is what is actually being realized in many fields. Agriculture field is not very well studied. And I request Dr. A.K. Singh to create a school of epigenetics. I have been trying to help a scientist, Dr. Suresh Kumar, there at IRI. Is not getting appropriate support. And I'm sure epigenetics must be mainstreamed. There's a lot more. As geneticists, we are preoccupied with genetics. We have the love for genetics. And we should not have blind love for genetics. We have to have our eyes and ears open. And
be proudly telling our scientists are very
march in the direction of embracing new developments and the process.
I just
creating new avenues for research. Manon Mani, kindly put off your mics. And I know epigenome is quite complex. Globally, a lot of efforts are happening, starting from Shakespearean concept of nature and nurture, John Locke's concept of blank slate, and then mind shaping the behavior and characters through nurture. They are all those 18th century, even 17th century stuff. Charles Darwin's concept in Origin of Species, as he has described, and subsequently Francis Galton's philosophy and nature versus nurture theory, and how we should be actually working in this direction now. And that is more important. Heritability. In twin studies are ranging from 21% to 67%. What it is? Recent study published in European Journal of Pain in 2013. It talks about epigenetics and its role, and how heritability, genetic plasticity, all that we used to discuss and describe, and stability or instability of genotypes, all those requires in-depth studies in epigenetics. Johansson's concept of genotype environment and genotype environment interactions. This is the time to delve deeper into genotype environment interaction, even genotype interaction. And I'm sure there is plenty of opportunity for us to build schools. How genotype environment interaction can be studied in great detail. I can cite few examples. And before that, let me tell you how nurture works. Nurture works through ionome. Nurture works through metabolome. Nurture works through hormones. Of course, through transcriptome and proteome. Nurture works through epigenome. Whether it is small RNA, whether it is histone modifications, whether it is other changes with regard to mobile elements. So all these as a component of epigenome. And importantly, not change in a single generation, but transgenerational epigenetic alterations. That has profound effect of phenotype. In a recent study, Dr. Suresh Kumar has demonstrated that how the epigenome changes if you grow a upland variety continuously in lowland, and again shift it back to low upland, and vice versa. A lowland variety or irrigated system variety grown several years under irrigated ecosystem, shifted to upland, and then back to again irrigated system. So how epigenome alters itself, how plasticity is inbuilt in certain genotypes, but not in other genotypes. A lot more needs to be studied, and a lot more to converge on. We understand only how DNA methylation works. A lot more with regard to other changes in the cellular environment is very, very poorly understood. So, environmental sex determination is one which is known for epigenetic control. Sex preselection of sheep embryo is also another example. Endosymbionts manipulating sex determination in insects. Another example how beyond genetics we must be really talking about and thinking about. Even in ferns, how environmental sex determination occurs. In ferns, not just animals or insects. Epigenetics has a link between nature and nurture. I believe we should be able to do that. So beyond genetics, you said the title was genetics and food production, but I'm telling genetics and beyond genetics and food production. How do we really do this? It's very much known that epigenetic programming by maternal behavior in human infants is very well known. It operates at a very subtle level. Breastfeeding of children and how cortical stress reactivity differs among children who are best breastfed and who are not. And what are the epigenetic alterations taking place? Maternal care affects DNA methylation at the CPG islands of glucocorticoid receptor, NR3C1 gene. This is a beautiful example of how epigenetics can influence human behavior and stress management in children. Maternal care gene expression is a very important component of child development. It's not just plant productivity. The whole societal development needs some, has to take care of, has to, understand the effect of genetics and epigenetics. So, maternal care gene expression, particularly the maternal leaking and grooming in case of rats and even other animals, experimentally demonstrated recently that how NR3C1 gene operates and how it works through epigenetic alterations. So friends, I can go on telling you such instances in animals, dairy animals included, manual and mechanical milking, poultry, birds, chicken cased and uncased, animals open field grazing and stall feeding. So a whole lot of areas are there for us to go beyond genetics for improving productivity, even may give more agricultural productivity. When I say agriculture for us, it includes everything, animal, fishery, and all that. So developmental plasticity is very important. Epigenetics during pregnancy is very, very important. I'm not going to those details. Nutrition and epigenetics. Today we talk of nutrigenomics. Nutrition and epigenetics. How the same gene behaves differently through epigenetic marks because of different nutritional situations, and how phenome differs by way of that. There are several examples of nutritional regulation of epigenetic modifications. And many genes which are there. And I would suggest one can go through the paper in Advances in Nutrition, published in 2021 by Kai et al., which describes so well the effect of nutrition on epigenetic modifications. And how even some of these epigenetic changes moves from generation to generation. And you can refer recent developments and how mRNA genes regulate various other genes in human. And these are all epigenetic modifications, and how we can harness these changes in case of plants. And in case of turmeric, for instance, epigenetic action on SDAC gene, which is an inhibitor. SDAC gene, which is an inhibitor. And that gives us polyphenol, more of curcumin. Similarly, in case of garlic, diallyl sulfide, again is the SDAC inhibitor. Green tea, DNFT inhibitor, that is the epigenetic action. And how epigallocate gallate EGCG gene gets modified. There are plenty of examples now in plants, how plant foods and how epigenetically active compounds and what happens. There are plenty of such examples. And how epigenetic marks can be also erased. What are the writers of epigenetic marks? Like DNMT, acetone acetyltransferase hat, and how SDAC erases the epigenetic marks. There are many such examples, and I'm sure we should be able to understand climate resilience more and more using epigenetics beyond genetics. So genetics and epigenetics should give us new dimensions to address malnutrition, to address climate change, and address sustainability. Dr. R.B. Singh talks about factor productivity. Factor productivity is declining. How can we address total factor productivity? Can we enhance nitrogen use efficiency, phosphorus use efficiency, potassium use efficiency, water use efficiency, lesser and lesser use of inputs, productivity not compromised, genetics and beyond genetics, epigenetics, playing the role, and appropriately mainstream and taken advantage of by my builder friends, geneticist friends, molecular biology colleagues, and I'm sure it will happen. I'm very optimistic. Only when you are optimistic, newer pathways open up. That is what I have seen during my lifetime. If you are pessimist, you will lose sight of those new ideas. And plants speak, Professor R.P. Sharma is to say. If you go to the plants, they will tell you the path. And if we close our eyes, if we are disinterested, we will not be able to discover something new. So let us open our eyes, be forward-looking, evolve continuously, and use the power of genetics and epigenetics to build a future, a positive future, a nature positive future, a sustainable future for every one of us. So thank you very much, Dr. A.K. Singh, for giving me this opportunity. So thanks to all of you, friends, for being here, all teachers, all colleagues, all students who are here. Thank you very much for being here. And thanks, Professor R.B. Singh, for giving me sufficient time, despite my bad throat. And thanks, Professor R.P. Sharma, for being there physically. What an abiding interest in genetics. Hats off to you. So thanks to all of you. Dr. G.P. Singh, Indian Society of Genetics and Plant Breeding, will partner this particular event. Thanks to every one of you. Thanks a lot.
Listening to you is always a delight, sir. Thank you very much, sir, for joining us online in spite of your ill health and blessing us with your enlightening inaugural address. We wish you a speedy recovery, sir. It's time to hear from our chairman. Sir, may I request you for your kind remarks.
Is it all right now? Fine, great. Dear friends, I'm sure you will all agree with me, such an outstanding presentation. Despite his bad throat, not so vigorous health, which he always enjoyed, he has given such a wonderful presentation. It's amazing. Friends, I would urge you for this effort to give him a standing ovation, if you may kindly, please. Professor Mahapatra, we cheer you for you and wish you your quickest recovery to the best of health. We need your best of health all the time. Friends, I have been asked to summarize, to summarize such a wonderful, great lecture. It's offering a fiat to myself. And this will be a difficult proposition. However, I'll try to pick out a few points which I think were emphasized and probably will take home messages. He started with the word that we must be able to build outstanding partnership and collaborative approach right from the plow to plate. And that includes the entire value chain management, all stakeholders along the value chain, we should have partnership. 2, he also underlined, and very importantly, that quality teaching is absolutely essential to bring out the perspectives which are necessary for sciences like genetics to develop further. We recognize thus that the Mendel laws will ever enrich biological sciences the way the impact has been described. So biological sciences, agriculture, you know what it means for the humanity as a whole, and therefore to give the thriving push to biology, we must be able to make the best out of what came from Mendel laws. Mendel chose, which is a lesson for all of us, to undertake disruptive ways of doing experimentation And he was not defeated by something which he did not understand. And he continued with the persistency to come up with innovations, which we must inherit. Of course, we are talking of the laws of inheritance. We must inherit this attribute also among ourselves. The concept that originated from there, the concept of alleles and allelism, is so very important that it goes through entire thread of heredity, as a matter of fact. We would like to see that the laws that he was able to bring through the use of mathematics underpins that appropriate data collection, numeration, and its treatment will lead you to some conclusions which are absolutely science-based, and you have to be guided by that. So the quality data collection must be, sometimes people guess and even criticize Mendel that how can it be so perfect that it comes always 3:1 or 9:3:1 or something like that. Now, I think despite all that, you can see the various adversaries and criticism. Using the varied systems, Mendel was able to develop models which boosted the future paths for the rules in biology. And I think this is very important, although Darwin contemporary, the kind of Darwinism effect on hiding or suppressing some of the values and beauties of Mendel laws were later on understood and explained by Mendel laws by itself. So I think it's very important to keep doing the right science and don't be disheartened. We have just seen the examples of how the proper science doing the genes which reduce the height and gave you the entire new plant type concept, the dwarfing types, whether it is the rice or wheat, RST genes or the WG genes we get in the two crops, which led to the development of what we see today as the basis of Green Revolution. As a matter of fact, I remember as I had my student for master's degree, a person known, I'm sure to each one of you, Dr. Ravi Prakash Singh, who is the head of the wheat breeding program at CIMIT, and you know, he worked with me on RST genes, the height reduction genes, RST1 and AS2, and gibberellin, effect of gibberellic acid. How will it influence in identifying the genes? And that was as early as, I think it was in before I left Banaras and the university. So, okay, between 72 and 79, must be somewhere 75 or so. This is where the work was done. So I mean to say that probably if we take some of those lessons from the various sources, we will be producing students like Ravi Prakash Singh, and of course many are sitting here, and they have done wonders in their own way. Also, I thought that the kind of mention which has been made, which needs to be highlighted, and congratulations to the institute, the director, and the concerned breeders. The high-yielding varieties of wheat, ST2967 particularly. VP Singh is here, we recognize that. Basmati 1121 and Ashok, Basmati 1509 and so on. All these have changed the entire scenario of India. And this is all based on Mendel laws. And how this, the germplasm analysis and the genes which are being searched through the process of evaluation, characterization, and using them for implementing or discovering new alleles and developments is absolutely essential. A discovery which, although we always mention about the rice and wheat, but the mention which Dr. Mahapatra made of the sugarcane variety, which Dr. Bakshi has developed, as a matter of fact, is amazing again. It's outstanding. It's changing the entire economy. If basmati rice, our rice export can bring us 30,000 crores per year, which is never done before, I'm sure this is all Mendel laws, which is allowing us to do that as long as we follow it vigorously and correctly. The allele discovery, which I think is the strongest message, has the strongest impact through mutagenesis and this position effect of the treatment of Nagina bice N22, which many of the variations which have occurred, and it has provided you, looking at the entire genome sequencing, how differences in positioning effects can really make the various combinations of characters. It was very important to note, Dr. Mahapatra, that haplotypes for breeding, particularly for brain traits enrichment, are the most important ways that we should go ahead now. Again, you mentioned the work of Charles Stewart, a colleague. He was my colleague. We went to school together at North Carolina State University. Professor Gupta, you know him. Charles Stewart, you visited him, and he had taken your on. Charles Stewart was the one who worked with the isogiants, and he brought in maize the breeding programs for quantitative traits through the molecular genetics, and he was able to, he was the first person to marry quantitative genetics, population genetics, with molecular genetics. You can see here, we have been doing work separately, but if we can see the two things together, which Charles Stewart demonstrated with Major Goodman later on, I think is extremely important. It's a message which Dr. Mahapatra also gave. We also found that cloning of quantitative trait loci, this is something, a very strong message. And after all, most of the traits that we worry about are quantitatively governed QTLs. And therefore, if agricultural production has to be credited to the kind that we have reached today, 6 times, 11 times, or so and so forth in the various commodities, from 1965 onward, 100 years after Mendel made his paper, first presentation of his papers. And if you look at that kind of development, it is because of the Mendel laws. It is again an enigmatic situation that despite all these developments, there is an Indian enigma. An Indian enigma and challenge I'm not going to repeat. At the last, Dr. Mahapatra was mentioning about the child health, child care, and so on and so forth. Do you know half of the world's wasted children are our own children? 35% of the stunted children of the world are Indian children. And if that is the situation, despite all kinds of revolutions we have, green, white, yellow, golden, you name it, the rainbow revolution, we have that kind of intensity of the underprivileged children. And how with a stunted soldier of a child, we can build a new India. We can't. Therefore, I think it's very important to see how best can we use the best of the science of genetics to really overcome some of these difficulties and paradoxes that we are facing. And for that, Dr. Mahapatra highlighted pulses in all seeds. The genetic gains plus the policy should be together. It is not, and he underlined, science is important, but equally important is policy for science to reach whom it is meant for. If you don't have the right policy, the discovery will never be delivered. And therefore, he asked for a match between the science of discovery with the science of delivery. And this, I think, is extremely important to bring together the private sector and the social responsibility of science with the corporate social responsibility together, so that very often Dr. Mahapatra mentioned about seed problem. The seed rate of replacement and so on and so forth will be taken care of. And that will promote PPP, the partnership between the farmers, public sector, peasants, and so on, so forth. He very often used the word disruptive technology transfer. Yes, we should be able to identify the disruptive technologies. We should be able to know that there is a different way to go about for those disruptive technology to be transferred. We need to discover the revolutionary genes, especially in pulses, IL sheets, through either create new ones through chemical mutagenesis or through genome editing or any of the new technologies that are available. And then impose along with that the agronomic management practices so that a blockchain technology development helps in carrying those discovered new combinations of genes to the farmer's field. Therefore, the artificial intelligence, digital platforms will become very important during this time and onward. We must be able to continue and support good work, strengthen genetics teaching in the universities for posterity. The rice, wheat, maize, sugarcane examples are there. And we should be able to follow this furthermore and more in many other crops, diversify the implications of these things. Of course, pulses and oilseeds were highlighted. The socio-economic, ecological, environmental, and the kind of agroecological factors which are important today should be included in our decision-making process. If Swarna has to be the global variety for India, we should be able to make sure that it benefits from the kind of gene editing programs continuously so that it doesn't fall down, so that new enrichment to that variety continues time and again. Now, I'm just giving a few examples of that kind. Therefore, if that be the case, sound genetics will help us reshape India to build new India. Finally, my dear friends, the last thing that Dr. Mahapatra highlighted, but it's not genetics alone, it is genetics plus. Nature and nurture. He talked of the epigenetics. To fight, I will say, just in one sentence, summarize all his work, all what he said in epigenetics is, the epigenetics is necessary to fight epigenetic fixation of stunted and wasted children of India. If you can solve this problem, you have solved the main problem of India. Do you know that these wasted, stunted children cost to Indian economy 10% of the GDP of India is lost every year because we have these many stunted and wasted children. So if epigenetics can help this problem, you help India in the best possible way. I will say all those aspects of metabolomics, microbiomes for changing the breeding pathways. Microbiomes can change the breeding pathways, as a matter of fact. And if those things could be included, we will be able to really have the resource management, soil enrichment, soil organ carbon economy, and the total environmental system to help you build a new India. Ladies and gentlemen, dear friends, this is what we got from Mendel. We should express our highest gratitude to that saint who, despite all kinds of problems, he was able to continue with determination till the last days of his life. Someone said, Mendel continued, though he was getting heavy and heavy in weight all the time. But he continued till the last day. Now, this is what actually is the lesson we learned. If we have to build new India, if we have to meet the sustainable development goals, if you have to fight the enigma of India, I think we should always take lesson from what Mendel did. And thank you, Professor Mahapatra, for your outstanding inaugural address. Thanks a lot. Thank you, sir. All the best.
Thank you, sir, for beautifully summarizing the presentation and presenting your insightful remarks. Moving towards the end of the program, may I now request Dr. Vinod, Organizing Secretary, to present formal vote of thanks.
Good afternoon, everyone. Honorable Chairperson of today's function, Professor R.B. Singh ji, former Chairman ASRB and former Director IRI, our esteemed Chief Guest and Speaker, Dr. Trilochan Mahapatra, DGICR and Secretary DR, Dr. E.K. Singh, Director IRI, Dr. R.P. Sharma, Chairman, 15th Genetics Congress Trust, Dr. J.P. Singh, President, Indian Society of Genetics and Plant Breeding, distinguished dignitaries, delegates, faculty members, and students. I am privileged to propose the vote of thanks on this important occasion of inauguration of the 3-day commemorative symposium on the birth bicentenary of Gregor Johann Mendel, the father of genetics. First and foremost, I would like to extend my heartfelt gratitude to our chief guest, Dr. Trilochan Mahapatra, for delivering this inaugural address on the very important topic, genetics and sustainable food security in India. The applications of science of genetics have led to tremendous increase in food production as well as in improvement in quality of food, leading to green revolution and subsequently food security in India and elsewhere. The sustainable food production depends to a great extent on the advancements in the genetics, leading to the development of suitable plant types according to the prevailing agroclimatic conditions. We are thankful to you, sir, for delivering this important inaugural lecture despite your ill health, and I wish you very speedy recovery. I would also like to thank my sincere thanks to Professor R.B. Singh for graciously consenting to chair today's event and for summarizing and giving his valuable remarks. The idea of celebrating 200th birth anniversary of Mendel was conceived by Genetics Club about 4 months ago. The time was short and enormous effort was required. Effort was made by almost everyone to make this event possible and memorable. Here, a special mention is required of our director, Dr. E.K. Singh, who not only guided us, but also made personal efforts to make this event possible. Though an elaborate vote of thanks is required to acknowledge the efforts of all, which I reserve for the later stage, here I would like to acknowledge the tireless effort of Dr. K.K. Vinod, Dr. S. Gopalakrishnan, Dr. Gyan Prakash Mishra, Dr. Firoz Hussain, Dr. Akshay Talukdar, Dr. V.S. Hegde, Dr. H.K. Dikshit, Dr. S.P. Singh, Dr. C. Bharadwaj, Dr. Shailesh Tripathi, Dr. Pralay Bhaumik, Dr. K.K. Mandal, Dr. Vignesh, and their team members, including students, for making this event possible. My sincere thanks to Dr. D.K. Yadav, ADG Seeds, and Dr. Ajveer, Head Genetics, for their help at various stages of planning. I also express thanks to Dr. R.P. Sharma and Dr. J.P. Singh for their support in organizing this event. Both 15th Genetics Congress Trust and Indian Society of Genetics and Plant Breeding readily agreed to co-host the event with Genetics Club. I extend my heartfelt thanks to Professor B.D. Singh from BHU, Dr. K.V. Prabhu, Chairman PPV FRA, and Dr. S.R. Bhatt from 15th Genetics Congress Trust, and Dr. C. Vishwanathan, Head Plant Physiology Division, who provided valuable inputs during the planning stage of this program. Thanks to Dr. Harita and her team for comparing the program, and Dr. Burman for providing his valuable support. I would also like to thank the distinguished dignitaries and former faculty members of genetics for gracing this occasion with their presence. My sincere thanks to Dr. S.S. Patil, Dr. P.K. Gupta, Dr. B. Mishra, Dr. B.M. Prasanna, Dr. N.K. Singh, Dr. Tandon, Dr. J.C. Rana, Dr. B.B. Singh, Dr. V.P. Singh, Dr. Gulati, Dr. Tumar, Dr. S.R. Bhatt is now he has arrived, he is present here, and many other faculty members and distinguished guests who are present here. Yeah, Dr. Velu is there. So I thank all of you for your presence, for your gracious presence on this occasion. And finally, I thank all the delegates who have attended physically, as well as those who have attended, joined this program online, faculty members, students for their participation in this program in a very large number. Once again, I thank each and everyone for making this event possible. I also thank our joint directors, Dr. Rashmi Agarwal, Dr. B.S. Tumar, and Dr. I.M. Misha, who are present here for their gracious presence, and directors of our sister institute, Dr. Shishane, Dr. Ashok Kumar Singh, and also Dr. R.M. Sundaram, who are present here. Thank you all for addressing this occasion. And I also thank Madam Renu Singh for her gracious presence on this inaugural function. Once again, I thank each and everyone for making this event possible. I hope the next 3 days will be fruitful and memorable for all of us. Thank you.
We have a photo session just outside the auditorium, so kindly join us for the session. And after this, lunch is arranged in the food court just behind the auditorium, and all the delegates are requested to join us for the lunch, and we'll assemble in the auditorium for the next session at 2 o'clock.